Enzymes work at an optimal range but can be denatured. When we start thinking about all the different things that can affect enzymes, we often think of pH, concentration, temperature, and the presence of inhibitors. So, when we have results that we could measure, we can often think of this as an experimental setup. So, when we brainstorm all of the different possible things that could affect how fast reaction rates happen with enzymes, we can then start manipulating variables to figure out how they affect them. So, if we were to think of this as an experiment, we would want to manipulate one factor at a time, and that would be our independent variable. When we isolate this one independent variable, everything else becomes constants or controlled variables. Now, controlled variables are not the same as a control group, where we have a baseline treatment for our experiment, and our baseline might be set up as a standard or what is normal. We can also have no manipulation, where you just remove the independent variable altogether. Or we can use possibly like a denatured enzyme, where we know we're going to have negative results because we know it's not going to work. Now, in addition to our control group, we have experimental groups. And again, we're going to want to vary the independent variable while keeping the constants constant. So if we are changing the concentration or the amount of the enzyme, we would want to still have the same temperature in every group. That would be a constant. We'd want the same number of starting substrates. We'd want the same pH. So all of those other things that need to stay the same in each trial of the experiment would be constants. Again, when we're talking rate of reaction, we're talking about the number of products produced divided by the time. So we call that products per second. And we then can analyze the trends as a result. So in this graph, it's showing that the rate of reaction, or how fast the reaction is going, will increase as the enzyme concentration increases. But then it starts to plateau here, and it hits a point where the reaction can just no longer go any faster whatsoever, and adding more enzymes will not increase the rate. However, adding more enzymes does not decrease the speed of it either, according to this graph. When we start looking at other enzyme graphs, we sometimes see that there are optimal conditions. Now, we have two different types of enzymes here, pepsin and trypsin, and it shows that pepsin has an optimal pH of 2, and trypsin has an optimal pH of 8. And they don't work outside of that range. So as we look at pH, to understand pH a little bit, again, pH is the concentration of hydrogen ions. And at neutral pH, or 7 of distilled water, you would have the same concentration of hydrogen ions and hydroxide ions. As something becomes very acidic, and let's say it has a pH of 1, like stomach acid, it would have 1 times 10 to the negative first hydrogen ions. And this is still a small number, but this is incredibly bigger than a pH of 13, which has 1 times 10 to the negative 13th. Well, we move that decimal place 13 places over to get the hydrogen ion concentration of bleach. So again, looking at this model, it's a little hard to understand, but understanding again this idea of lock and key. So let's say we have all these hydrogen ions inside our stomach. So we have this protective layer of mucus that's preventing these reactive hydrogen ions from damaging the cells that make up our stomach. So what we're imagining here is that these individual cells of the stomach lining are going to make mucus, but then some other ones are going to make what we call pepsinogen. Not pepsin, but pepsinogen. So at this point, there is something sitting on the active site which makes this inactive. So you have proteins that you make through your genetics, through transcription and translation, within these cells. And we don't want this enzyme here breaking down cells that our own genetics have made. But we do want it to work to break down proteins that we have eaten. So as pepsinogen is secreted into this gastric acid, it reacts with the hydrogen ions, and which then turns it into an active form of pepsin, which can then break down the proteins and continue the chemical digestion that's taking place within your digestive tract. Again, these are happening specifically in different locations. Pepsin is located in your stomach, and it begins the process of protein digestion. However, trypsin is made by your pancreas, which also makes bicarbonate, and the bicarbonate acts as a buffer and turns the pH to almost 8 here in your duodenum. And it's in your duodenum that trypsin then takes over the protein digestion, and it's going to work in this environment. So there are optimal environments and that specificity of things working in different locations. When it's not working, it's often denatured, which means the shape has changed, and pH can do that. pH can take pepsin and you get it outside of the range, and you get it into pepsin is still going to be traveling through your digestive tract, but once it hits the duodenum, it's no longer going to work because it doesn't have any ability to work above a pH of 4. And the same thing, if trypsin were in a different location outside of this range, it wouldn't work. We often think of denaturing not just with pH, but often with temperature. If you get things too hot, you change the shape of the protein. If you change the shape, you change its function. So going back, thinking about tertiary structure, when we get to the tertiary structure, the functional three-dimensional shape of the protein, we have R group interactions. The secondary structure working backwards is connected by hydrogen bonds. But primary structure is connected by peptide bonds, these strong covalent bonds. So a lot of times, even if you get really a pH that's way out of range from what's optimal, or you get a really incredible high temperature, you may denature these shapes, you may change these shapes, but a lot of times you won't actually destroy the primary structure. So therefore, sometimes denaturing can be irreversible. It can't fold back into the correct shape, and sometimes it can still fold back into a functional shape because you haven't destroyed the primary structure. Now again, this idea of optimal for temperature, optimal is most of the time going to be whatever the body temperature is of the organism that it comes from. So through evolution, things have been designed and have worked optimally in different locations. The optimal temperature for an enzyme found inside a human may not be the same optimal temperature for an enzyme found within a bacteria from a hot springs. So other things that can affect enzyme function are inhibitors. We have competitive inhibitors, and again, we have this lock and key model, the active site where the substrate lands. But a competitive inhibitor will occupy the active site, and therefore the substrate cannot bind, and therefore the chemical reaction cannot occur. We also have non-competitive inhibitors. So we have an active site, but we also have this allosteric site. And again, the substrate is going to land on the enzyme, and enzyme 1 is going to turn substrate A into B. And B is going to interact with enzyme 2 as the substrate and turn into C. And C would be the product, but then it becomes the enzyme or the substrate for enzyme 3. And this continues and continues and continues until we get a final end product. And sometimes this, again, having enzymes be active at certain times and not other times is an important aspect of enzymes. So if we get this end product, we no longer need the end product. We don't necessarily need to continue this process. So the end product could bind to the allosteric site, which then changes the active site shape, which allows the initial substrate to no longer be able to bind. So again, these types of inhibition can be reversible and irreversible depending on the situation. So there is an optimal range for enzymes. Denaturing can occur, and pH, the concentration or amount of substrate and product, temperature, and inhibitors can all affect enzymatic activity.